Good afternoon, everyone.
Hello.
Hello. Good afternoon.
All right. Welcome back and thanks for coming out today. Hope you're doing well. I know there's sort of an energy about finishing the semester, right? Just get it done with so we can go on and enjoy our holiday break. So hang in there. One more week of lectures. Congratulations on getting to your final week of lectures. You know that a week from this Thursday, you'll be taking your final exam, right? Final exam is scheduled for Thursday, December 16th at 12:30. Thursday, December 16th at 12:30 is your final exam, okay? So let me just ask a question quick in the chat window. Did you receive the final exam study guide that was emailed to you last week? Okay, so it's my hope that it is in your possession, even if you haven't started preparing, that at least you're aware that it exists. Okay, great. So I see a lot of yeses in there, and that's wonderful. Okay, folks, we're going to hit chapter 15 today hard, definitely finish it. Maybe start chapter 17. We'll see how this goes. We're just going to rock and roll. So do you have any questions before we get started? Okay. All right. Well, let's begin then. So I'm going to share my screen with you. Okay. And the last time we were together, we were talking about this topic. Factors affecting the likelihood of disease occurring. This is a topic that comes right out of the first few pages of chapter 15. Okay. And so, if you have the same book I do, I'm basically on pages 424, 425, 426. And so we defined two factors. Number one was the type of portal used, understanding that microbes prefer to enter the body through a specific route. And if they're allowed to access the body through a specific route, then they're more likely to cause trouble. Number two, the number of pathogenic cells or virus particles that enter the body. Then we went on a little bit of a tangent here talking about infectious dosage, right? ID 50. We finished up talking about lethal dosage, LD 50 as our example. Okay, so that's where we finished. That's where I'm going to pick up with my next note taking. Okay, so right here I have the same topic. I put the first two factors in. I'm just not repeating the content that you have for infectious dosage and lethal dosage. Okay, so the third factor that impacts whether you're likely to develop a disease is this idea of adherence. Basically, can the microorganism in question stick to a body surface? If a microbe can attach itself to a body structure, if it can grip to it, it is much more likely to cause trouble over a period of time. Okay. So adherence, most pathogens have the ability to stick to a number of body structures. Okay, again, if the microbe can stick, it's more likely to cause you harm. Okay, so this references figure 15.1 in your textbook. All right, and you can see panel A of 15.1 showing you surface molecules, which are called adhesins, okay, that are being used to bind to some kind of molecular structure in the body. You see panels B there, artificially colored E. coli cells in the urinary bladder. And letter C there, the bacteria, which are artificially colored purple, adhering to human skin, okay? So I don't think we're saying anything terribly novel here that you wouldn't have appreciated before. But think about, really, particularly in the medical community, the importance we put on washing our hands before a surgical procedure, right? We want to de-germ that surface before a surgeon touches the patient's body, okay? And so related to this idea of adherence is this concept of biofilms, okay? Okay. A biofilm is really a community of microorganisms that are living together on the surface, on a surface of your body. That surface could be the wall of your urinary bladder. It could be the wall of your urethra. It could be the surface of the enamel of your teeth. That's your plaque, right? Dental plaque. That's a biofilm. And there's some increasing evidence to suggest that at least some cases of atherosclerotic plaque inside your blood vessels might actually be biofilms as well. And that's kind of a disturbing conclusion because that's going to be hard to deal with. Okay, so biofilms represent communities of microbes that support each other as they live, as they attach to a surface of the body. Okay, so examples would be, dental plaque, film in the wall of the urinary bladder, etc., etc. Okay, so biofilms are actually a real concern, not just in general, but particularly to the medical community, because we're starting to appreciate that, things that we started, bacteria that we started to believe were simply single cell creatures that are, roaming around the body every once in a while and sometimes cause harm. Actually, it seems that the majority of the lifetime of microbes in your body are living in community with each other. Think about the idea of community from a human perspective, right? Community from a human perspective, an organized group of human beings, society, allows that society to offer features, characteristics, and properties to individuals that individuals by themselves would not able to have access to or appreciate, right? I mean, who among us as an individual could go pump oil out of the ground, refine it, and have gasoline to run our car? Who among us as an individual could actually engineer, manufacture, build, and assemble a car, right? None of us. We could not do that as an individual. I mean, maybe if we want to take the rest of our natural life to dedicate ourselves to that, we could. But my point is that working as a community, as a team of humans, we now have access to cars and farm equipment and machinery and things that allow us to do things, for better or worse, that we couldn't do as an individual. And we're starting to appreciate that the very microbes that colonize our bodies are also living in community with each other. Okay, so what I wanted to do is just show you a quick video on biofilms, just simply as a way of getting you to appreciate this a little bit more, give you some visualization that your book can't give you. Okay, so I've linked a YouTube video up here. It's about 3 minutes long, and we're going to watch this just to provide a little bit more context about the importance and significance of biofilms. Okay, so let me cue this up. Okay. All right. Okay, so here's a film. Admittedly, it's about 9 years old, but that doesn't mean the information is incorrect. The information is extremely correct, and it presents the information in kind of a quirky, fun, a little bit of a gross way, okay, as many things in micro are. So let's have a watch over this and see what it can tell us about biofilms. Here we go, folks.
Hello, I'm Hank Green. Welcome to SciShow, where we love slime. Slime is great, except when it's the wrong kind of slime. And when I say the wrong kind, I mean the kind that can kill you. Biofilm, also sometimes referred to as slime, is a type of bacterial colony that produces a sticky organic glue which anchors the microorganisms to whatever surface they fancy and also to each other, making them exponentially better/worse than they would otherwise be. It's also the latest addition to my list of things I wish I didn't have to worry about. We tend to think of bacteria as freewheeling loners making their way through the world all by themselves, and sometimes they are that way. Those are called planktonic bacteria. But scientists are beginning to realize that bacteria actually spend most of their lives as biofilm, which makes them way better at doing what they do, whether it's helping us digest food or killing us with cholera. Just as living in towns and cities makes us more effective as a species, bacteria living together as biofilm allows them to divvy up labor, protect each other, and share food, and that frees up more time and resources to do the stuff like digesting and the killing. Their secret is the slimy goo that many bacteria secrete, made up mostly of complex sugars called polysaccharides, along with some proteins to form this sort of infrastructure of these bacterial towns. This concoction of chemicals is actually very similar to a substance our own bodies create, and that you may be familiar with: mucus. Through this slime, different bacteria can share nutrients in water. They can also send signaling proteins to let them communicate. They can even swap DNA to pass along their genes. It's like the United Freaking Nations of germs up in there. Because this structure makes microorganisms so successful, scientists estimate that 60 to 80% of the bacteria that cause human infection actually live as biofilm. And because the only thing they need to start a colony is water, they pretty much form in every environment on Earth, from super hot springs to the inside of your mouth. In fact, if you rub your tongue along your teeth right now, you can probably feel, maybe even taste, the most widely known form of biofilm, plaque, which is actually a biofilm that contains up to 500 different species of bacteria and is the #1 cause of periodontal disease. Other biofilms are responsible for even gnarlier infections like cystic fibrosis, Legionnaire's disease, and all kinds of chronic ear, sinus, and skin infections. To make matters worse, their thick, slimy matrix often makes biofilms antibiotic resistant. One study found that bacteria in biofilm were 1,000 times more resistant to antibiotics than the same bacteria were when they were on their own. But the real insidious part is that biofilms periodically shed their individual bacteria, which then go off and start their own infections. While antibiotics can kill these planktonic bacteria, often making everything seem cool for a while, the biofilm Death Star is just hanging out there waiting to release more of its little TIE fighters to attack the body again. As a result, biofilm-infected tissue sometimes has to be removed surgically. They're that tenacious. So I know it's gross, and also it's scary. So what are scientists doing to protect me and also you, but more importantly me, from biofilms? Well, for now, scientists are finding out more about them and how they work so that they can develop drugs that can break down the slime in order to help the antibiotics work at the bacteria within the film. And what are we doing here at SciShow? Well, just know that we made a conscious decision to not show you any actual images or footage of biofilms in the human body. So be thankful for that, and have a good day. And if you want to keep getting smarter with us here at SciShow, you can go to youtube.com/SciShow and subscribe.
Okay, folks, so a little bit of a lighthearted coverage of a very serious topic, right? Biological films. Okay. So, as I prepare to continue to take notes, do you have any thoughts, questions, or statements about biological films? Okay. I mean, when you have an antibiotic-resistant infection in the body, you yourself now know of several strategies that you had to list out, on your exam of how microorganisms become resistant to an antibiotic. But one of those really was not to develop a biological film. And that's something that's only coming into focus in the past 10 or 15 years, which is recent science. Okay. So if you have a resistant microbial infection, I keep saying microbial, we think bacterial, right? But it could be fungal, it could be protozoal, we just don't know. And you're trying to treat this thing with a traditional antibiotic and you think, man, why isn't this infection getting better? It might be because the antibiotic is absolutely very good at killing a microbe, but it can't actually physically get to the microbe because of this film of material that is manufactured and secreted by the community of bacteria living on that space. It's basically like you having slices of peaches that manufacture their own jello. When you have jello with fruit bits inside, you can buy the jello with the fruit inside at the supermarket. What if the fruit bits were the bacteria and they literally manufactured their own jello? That's what the bacteria are actually doing here. And so they're basically creating a substance that protects them and walls them off from not only cells of your natural immune system, the white blood cells, but also chemicals such as antibiotics we're shoving into the body. So next generation antibiotics, scientists and companies, they're researching, well, how can I package an antibiotic with perhaps a concoction of enzymes where the enzyme would then nibble on the composition, the carbohydrates and so forth that are part of the biofilm, and break that down a bit more so that the antibiotic can gain better access to the bacteria that are part of the microbial film, okay? And so, long story short, I have a good friend of mine that recently went through sinus surgery. He had a bad sinus infection for many months, went through several rounds of intensive antibiotic therapy, and nothing was touching this thing. And finally, the doctor said the only option to deal with this infection really is a surgical option, where we go in and we scrape the internal sinus cavities. We scrape them and physically pull that film off of there, right? So it's not a pleasant thing, right? Okay, Samantha, thank you for sharing that. I appreciate that, and I hope you had a good outcome to it, okay? So biofilms are a big deal, and you're going to just hear more and more about them. You're going to see that medical science is focusing on biological films as problems with dealing with infections. Okay, folks, let's continue here. Next talking point. Okay, next talking point. I want to list out strategies listed in your textbook by bacterial pathogens to hide or penetrate from your body's natural defenses. Now, we haven't talked about your body defenses yet. We're going to do a little bit of that in chapter 17, so bear with me here. Strategies used by bacterial pathogens to penetrate host defenses. Okay, this is a major heading of text in my book. It's at the bottom right corner of page 427. So somewhere, if you have the digital text, you should be able to see a heading that says how bacterial pathogens penetrate host defenses. This is what I'm talking about at this time. Okay, I'm not going to talk about everything, but I'm going to talk about a few of these creatures. Okay, first is this idea of capsules. Okay, now I'm not talking about the kind of capsule you take and swallow medication here. We're talking about the kind of capsule that you might recall from lecture exam 1 content when we started the course and we defined the anatomical composition of a typical bacterial cell. And remember, we had that creature, that structure called a glycocalyx. And there's 2 kinds of glycocalyces. One is a capsule, the other is a slime layer. Okay, so recall that a capsule is a type of glycocalyx that covers some species of bacteria. All right. It seems to be the case, scientists are noticing that many pathogenic bacteria seem to have capsules. So in other words, the presence of the capsule seems to correlate with pathogenicity, pathogenesis, the ability of the microbe to overcome your body defenses. Okay, and so scientists are still working on why would this be? Well, there's a couple things that have been discovered. First off, obviously the chemical composition of the capsule varies from one species of pathogen to another. So there's not one single chemical composition we could talk about. But we do know that there's a lot of carbohydrate in the capsules, and those carbohydrates vary from one species of cell to another species of cell. Long story short, what scientists have discovered is that the capsules present on pathogenic bacteria often seem to interfere with the process of phagocytosis. So, depending what other biology you've had, that phagocytosis is basically when one cell eats another cell. So the idea here, would you have a human cell of your body that goes out and tries to eat bacteria before the bacteria can cause harm? Well, the process of eating the bacteria is interfered with by the chemical composition of the capsule. Something about the chemical composition of the capsule is blocking or at least slowing down the process of phagocytosis. Additionally, the chemical composition of capsules is such that some of the molecules that make up the capsule seem to have a structure, a shape, a geometry, which resembles, meaning it mimics. It's very, very, very similar to self-identifying antigens we find on our own cells. And by that, I mean, for those that know something about your body, I'm talking about MHC proteins. Some of the molecules found in the capsule have a structure that resembles molecules naturally found on our own human cells. And so the idea here is that when the human defense cells approach a pathogenic species of bacteria, the human defense cell might look at the chemical on the bacterium that resembles a natural molecule on our own cells and figure, oh, that bacterium, that cell, that thing that I'm investigating, it seems to belong in the body because it has a molecule that I recognize as good, as friendly. Okay. Some molecules that make up the capsule resemble self-antigens found naturally on host, I'll say human cells. These molecules confuse white blood cells into not attacking them. Okay. These molecules confuse white blood cells into not attacking them. Okay. All right, so capsules are a problem. And, as we deal, there's not going to be a single magic bullet that is able to disrupt the capsule of every pathogen, since the chemical composition of pathogens varies from one species to the next. So there's going to be a lot of hard work in the years ahead to develop specific drugs that can target specific capsules' chemical compositions. Okay, thoughts or questions? All right. Let's go. In the book, I'm choosing to skip over cell wall components at this point. I would like you to read it. I'm just not going to lecture on it. I am going to talk to you a little bit about enzymes. Okay. So, enzymes, obviously that's a generic statement. There are many, many kinds of enzymes in cells and even on the surface of cells and even outside the cell doing work. So which specific enzymes are we talking about here? let me give you an example of enzymes used by bacterial pathogens to help penetrate our host defenses. Okay. Okay, coagulases. Coagulases, okay. Coagulases are enzymes that are manufactured through the process of gene expression that you learn about in this course in bacteria, and then the bacteria secrete. They pump those enzymes out of the cell. Well, the enzymes do something inside your body. What they do is they promote the formation of blood clots. Okay. These are proteins made by bacteria and secreted into the host bloodstream to promote the formation of blood clots. I mean, hearing the phrase blood clots carries an ominous tone here, okay? But there's a certain weird logic for what the bacteria is doing. Okay, so let me ask some questions in the chat window. So you know what I mean by in your car, you have airbags, right? The purpose of an airbag is to protect you during a collision so you don't get hurt, right? So here's my question in the chat window. How many airbags are in the car you drive? How many airbags are in the car you drive? Okay, all right. Very good. Okay, thanks for participating. I'm a little surprised that everybody's got 2 airbags. That's how many I have in my car because I drive a 17-year-old car, which should be warning to you to never become a teacher. Okay. So, all right. So Elena has 4. All right. So Elena seems to have most airbags in our group. Here's the general logic. Okay. If you think about human beings sitting inside a car, the more airbags that you have enveloping you, surrounding you, hopefully the more protected you are from collisions, right? So those of us that have 2 airbags, well, that's better than nothing. 4 airbags, a little bit better. There are some luxury sedans, I think, that probably have 8, 10, 12 airbags, but those are for the rich of us in our communities. So what I'm saying here is the coagulases that are released by these pathogens are promoting the formation of blood clots because the bacteria want to hide inside the blood clots. That's the purpose. Okay. Some pathogens seek to form blood clots in the host, so the pathogen can hide inside the blood clot. You think about that. That's kind of a sneaky little strategy that Mother Nature's come up with. Oh, okay, let's take the infected host and cause its body to form little clots, and I can hide inside that clot like a human being hiding inside the interior of a car that's protected and in an airbag shell, in an airbag cage, right? So if the bacteria can hide inside the blood clot, that means white blood cells, B cells, T cells, macrophages, natural killer cells, the whole 9 yards, these cells cannot get at the bacteria. Okay, so it's a way of hiding from the defense cells. All right, so that's one kind of enzyme that is found in some species of pathogen. I'll give you 2 more examples of enzymes. First, we have hyaluronidase. Okay. Hyaluronidase is an enzyme. It's an exoenzyme. Okay. And so, you know, I expect that you understand it's an enzyme made by the pathogen, pushed outside the cell. That's why it's exo, to do its job. This is an exoenzyme which breaks down hyaluronic acid. Okay, so I have a question that I'm going to put in your notes, but I'm going to ask if you can try to contribute to the conversation in the chat window. Okay. Where in the human body, and it's not just the human body, it's the animal body, do you find an abundance of hyaluronic acid? Where in the human body do you find an abundance of hyaluronic acid? So I know I've got some A&P graduates in here, so I would expect you to do that. I'd expect you to know the answer to that. Okay. All right, so thanks for those that are putting some guesses in the chat window. You definitely have hyaluronic acid in your skin, no question about that. You would have some hyaluronic acid in some anatomy of the eye, no doubt about it. Okay, so you did a good job. You have the generally correct idea. So let me focus this a little bit. Yeah, absolutely. Now we're starting to get it in the chat window, right? Hyaluronic acid is a molecule. It's part of what's called the ground substance of the extracellular matrix of connective tissue. Okay. This is a molecule found in the extracellular matrix of connective tissue. Okay, so we won't go into an anatomy conversation right now, but you can appreciate that the purpose of connective tissue is to connect, to bind tissues together, right? And so anywhere you need to link epithelium to, you know, connective tissue, or, you know, anywhere you need to glue body structures together, you're going to have connective tissue being part of that anatomy. So it's throughout your body. Skin is a beautiful example, right? Because, you know, if you're at home, go to your bathroom or kitchen and pull out your skin cream and look at the chemical compositions. If you're buying a pretty decent quality skin cream, it's likely to have hyaluronic acid as part of the chemical composition of the skin cream you've purchased. Okay, so bacterial species will make this enzyme called hyaluronidase to chew away at the hyaluronic acid. So hyaluronic acid is actually a food. It is a nutrient for some species of pathogen. And if you think about what connective tissue does, it glues things together. You now have a pathogen's enzyme which can disturb the glue. And the reason for disturbing the glue is to allow the pathogen to literally burrow through your anatomy, to burrow like a gopher burrowing through the soil out front of your home. This enzyme will allow the pathogen to burrow through your anatomy to evade your body's defenses. Okay. You know, and I really mean burrow, folks. I mean, think about an apple and how you might have an apple and you might have a worm that literally burrows from the surface of the apple through to the core. That kind of burrowing can happen with some species of bacteria. And the ones that I'm talking about are ones that are typically labeled as flesh-eating bacteria. Flesh-eating bacteria often produce hyaluronidase. And of course, none of us really want to come into contact with that kind of bacteria because it's very, very, very serious. Okay. Questions? Okay, one more enzyme I want to share with you, and that's a type of enzyme called an IgA protease. Here's a question coming into the chat window. And here it comes. Are any of you old enough to remember IgA supermarkets? Okay. All right. So some of you are, some of you aren't. That's typical. Okay. All right. So at any rate, you know, a little bit of levity there. This topic is about IgA, but I'm not talking about a supermarket. And of course, if you don't know what I'm talking about, then the humor is lost on you. It's kind of like everything I tell my 7-year-old son. He doesn't get anything I say. So I'm just old. Okay. So IgA proteases are immunoglobulin class A enzymes. Ig for immunoglobulin. Okay. Immunoglobulin class A proteases, enzymes. Okay. And so just to give you a little bit of context here, okay, one of the ways that your body defends itself from species of bacteria is to generate a whole bunch of different enzymes. We'll cover a little bit of that next class, but the category of antibodies that your body manufactures called IgA has the ability to interfere with adherence. It has the ability to interfere with adherence. And again, if a bacterium can stick, it'll cause a problem. So one of the ways your body tries to defend itself is to manufacture IgA antibodies which prevent adherence. Well, Mother Nature's sneaky. As the bacteria realized through biological periods, through evolutionary time, as the bacteria realized that it was having a hard time sticking to surfaces, it started to evolve new strategies to counter that defense of the host body. Long story short, IgA proteases are enzymes that cut up the host IgA antibodies and thereby blocking or preventing the host strategy of preventing adherence. So the IgA protease prevents the preventer, okay? These enzymes interfere with host IgA antibodies, okay? So remember, an immunoglobulin is another name for an antibody, okay? These enzymes interfere with IgA antibodies. IgA antibodies block bacterial adherence. So these enzymes restore the ability of the pathogen to stick to a body surface. I mean, this really is biological warfare. I'm not talking about biological warfare from the perspective of human beings using biological material to terrorize each other. I'm not talking about that. I'm talking about the cells of your body doing something to interfere with the microbes in your body, and the microbes responding to that to prevent the initial strategy from being effective. That's what's happening here. This is literally biological warfare within your body. Okay. All right, folks. So there's some quick examples, 3 examples of enzymes that are kind of interesting. Let's continue our conversation. Number 3 here, antigenic variation. You already know this one. I know you know it, even if you don't know you know it. Okay. Antigenic variation refers to the ability of a pathogen to alter its outward appearance, okay, by shuffling segments of DNA around in a way that I hope I'll show you a little bit about next lecture as we end our course. Bacteria can shuffle segments of DNA, and when they shuffle segments of DNA, they get different proteins. Different proteins can be put out on the surface to give their external surface a different appearance, a different look. That's like me looking like I look now. And then on Thursday, I pop into class and I have a hat on, and I've drawn a mustache and beard on myself and different clothing so that I look different. Okay, so antigenic variation refers to the pathogen changing its external appearance, which confuses host defense cells. Okay. Shuffling of DNA segments in the pathogen. Shuffling DNA segments in the pathogen allows the pathogen to produce molecules that are different on a continual basis. And by putting these different molecules out on the surface, you can confuse the host defenders. Okay. This is one strategy that some of you answered during exam 2 when I asked you to talk about ways that HIV can evade the immune system. HIV can change its surface molecules, as most viruses can. Okay, but bacteria can do it too. All right. So there's lots of strategies that pathogens take. There's lots of strategies that the cells of your own body take to try to defend itself, and there's a continual tug of war. If you're a healthy person, not fighting an underlying health issue or genetic condition, you get adequate nutrition every day, you're resting well at night, you're hydrated. You know, we have the luxury of generally staying healthy for 6 or 7 decades of life, and then you hit your 40s, you're going to notice your body changing. You hit your 50s, you notice it changing, and it just gets worse the older you get. Okay. It's called aging, and there's not too much we can do about that. Okay, one more thing here. Strategy number 4, penetration into the host. This one, you have an interesting figure 15.2 in your textbook. Okay, here's the basic idea. Some species of pathogens, and I'm talking about Salmonella, obviously that's figure 15.2, but some strains of E. coli have also been identified that do this. What they have the ability to do is secrete these exoenzymes called invasins, INVASIN. These invasins bind to a host cell like one of our own body cells, and it triggers a chaotic reorganization of the host cell's cytoplasm. Okay, so exoenzymes called invasins are released by some pathogens which bind to host cells and trigger a random / chaotic reorganization of the host cytoskeleton. Now, let me ask you a question in the chat window. First off, first statement. Imagine a balloon with 10 swords inside the balloon, organized in a way that doesn't pop the balloon. Now, imagine you bump into the balloon harshly, causing the swords to jiggle around and reposition themselves randomly inside the balloon. Inevitably, if the swords start moving around in a chaotic fashion inside your balloon, what's going to happen? The balloon's going to pop, right? It's going to pop. Well, in terms of cells, if you imagine the balloon as a cell, the cell kind of pops. And what I'm saying here is that you get a tear in the plasma membrane of the host cell. And when you get a tear in the plasma membrane of the host cell, part of the cytoskeleton, remember the cytoskeleton is made of these polymers of proteins called intermediate filaments, microfilaments, things like this, these chains of protein. These filaments pop through the plasma membrane and puncture the cell. That creates a hole, a gaping hole in the plasma membrane that is big enough then for the bacteria to get on top of the cytoskeleton tube sticking through the plasma membrane and literally walk itself inside the interior of the cell. Okay, it's kind of sneaky. You force the cell to pop itself, and then you hop on the cytoskeleton and literally crawl into the interior of the host cell. Okay. Okay, so let me just read my notes to make sure I'm communicating correctly here. When the host cell damages itself, in response to the invasins, tubes of the cytoskeleton poke a hole in the plasma membrane, allowing the bacteria to walk along the cytoskeleton into the interior of the cell. This phenomenon, where the host cell chaotically reorganizes itself, is called ruffling, and you see that in figure 15.2. Okay, literally the cell is kind of flopping around like this as the cytoskeleton reorganizes itself. Okay, so unfortunately there are a number, if you think about these things, they are intellectually curious. They're kind of interesting what happens, but that's about as positive a statement as I can make, because after that, now the bacterium, the pathogen, is inside your cell, and it has access to the interior organelles of the cell, right? Has access to lysosomes, has access to mitochondria, has access to the nucleus potentially. It's causing problems, okay? All right. So, okay, we need to keep rolling here, folks. please let me know if you have questions. Otherwise, I just want to shove notes into this document so you have them for study purposes. Okay. Well, we're gonna shift gears here from strategies used to penetrate host defenses to strategies used to damage host cells, to actually cause real harm. And you could argue that the invasion strategy is certainly harmful. So these two topics are intimately related to each other. Okay, so in my book, this is a major heading on page 430, where it's titled "How Bacterial Pathogens Damage Host Cells." All right, the first and rather interesting one is where bacteria actually compete with the host cell for nutrients in body fluids, most notably the bloodstream. Okay, so the pathogen may compete with host body cells for nutrients in the bloodstream. Okay, that's like, that's kind of like you buying all the good food you enjoy, putting it in your refrigerator, and some random person comes off the street, walks into your kitchen and takes it out of your refrigerator and uses it, eats it. I mean, you would be upset if that happened, wouldn't you? I would. So that's what's going on here. Bacterial pathogen sometimes eats the nutrients in the bloodstream that our own cells need to survive. Okay. And a traditional and classical example of this is highlighted in figure 15.3. In terms of something called a siderophore. Okay, siderophores are manufactured by several bacterial pathogens, and what the siderophores do is allow it to bind up free floating iron that's in the bloodstream. For those that have had AMP, you appreciate that iron is super important for a lot of body processes. Most notably, you need an iron ion in the structure of hemoglobin to bind to oxygen, to deliver oxygen to your tissues and organs. So if you're low on iron, I mean really low on iron, you might actually be anemic because you can't bind the oxygen and deliver it to your tissues. Used by some pathogens to bind free iron atoms. This means that iron is not available for host processes, such as delivering oxygen to tissue, which you can appreciate is a very important process we really don't want to be disrupted. Okay. Let me tell you a very quick story here. Several years ago, one of my colleagues on staff at HAC, her son, 17, 18-year-old man, became, very fatigued and tired, didn't seem himself. So she took him to the doctor, and the doctor noticed that he was anemic. And of course, when you're anemic, people start worrying about blood disorders, various forms of blood cancers, things like this. So long story short, this 17, 18-year-old guy went through a lot of lab tests over a period of several months to try to figure out why he was anemic. All the tests came back negative or inconclusive. There wasn't anything meaningful coming back with the exam results. So they couldn't figure out why this 17-year-old man was anemic, and a healthy 17-year-old guy is about as healthy as he is ever going to be in his life. So, this thing really should not have been happening. And so after several months of attempts to explain this, the family physician threw up his hands and said, I can't explain why your son's anemic, but I'm on a hunch I'm just going to treat this with an antibiotic. There was no evidence of a bacterial infection, but let's treat this with an antibiotic. So long story short, 2 weeks after the antibiotic, the young man's feeling a lot more energized, has a lot more energy, feeling a lot better. Apparently, this young man had a subclinical bacterial infection of some kind that was resolved when he took a treatment of antibiotics, a broad spectrum antibiotic of some kind. And so the point I'm trying to make is, there are scenarios where, even the best intentioned doctors are going to misidentify what the cause of is. When you think anemia, you don't typically think about a bacterial infection, but it very much can be. Okay. All right, let's continue. Number 2. I'm going to go with number 2, production of toxins. I'm going to skip around a little bit for time's sake. Production of toxins in the body. So first off, we'll start with the definition of toxin. A toxin is any poisonous substance that is manufactured by a microbe. So here's a question for you in the chat window. Would it be appropriate to call antibiotics toxins? Okay. You see that we have some disagreement early on in the chat window here. Okay. So the idea that I'm trying to get to tickle around in your brain is that, it depends on context, right? I mean, if we are taking an antibiotic to cure ourselves, we're very happy with it. But obviously the pathogen inside our body that's going to be killed by this substance is not happy with it. Okay. So, from the pathogen's perspective, that's being killed by the antibiotic that's helping us return to health, that antibiotic is most definitely a toxin. It definitely is a toxin. And don't forget that, I'm responding to your comment, kid. Don't forget that antibiotics are Mother Nature's invention, not human beings' invention. It is true that we do manufacture some antibiotics, but the starting point for those antibiotics always came from Mother Nature's inspiration, right? Think about penicillin being made in the mold Penicillium chrysogenum. Okay. All right. Okay. So, I would argue that an antibiotic really can be considered a toxin. It happens to be a toxin that works to our benefit, but it is killing something, right? So, all right, let's talk about exotoxins. There's basically 2 major categories of toxin. An exotoxin is like an exoenzyme, right? It is a protein made inside the microbial cell and then secreted out to do something, right? Okay, an example here, one of my favorite examples is Once again, botulinum exotoxin, right? We did talk about that last class when we were talking about infectious dose and lethal dosage topics. Botulinum exotoxin is able to kill because it blocks the release of acetylcholine at neuromuscular junctions. Okay, botulinum exotoxin is able to kill animals by preventing the release of acetylcholine, which you know is a neurotransmitter. And long story short, without going into anatomy topics, you appreciate that for you to breathe, which is to say for you to ventilate, to inhale air, your diaphragm, which is a skeletal muscle, basically in the middle of your chest, separating the thoracic and abdominal cavities, that muscle has to contract and move down to change air pressure so you can bring air into your lungs. Well, that can't happen when botulinum exotoxin is floating around in your body, okay? So you can't contract the diaphragm, you can't bring air in, basically you suffocate to death, you cannot breathe, okay? So obviously you don't want this beast because it's really nasty, okay? Botulinum exotoxin is made by the bacterium called Clostridium botulinum. Okay, and as we said before, it's very toxic. You don't have to know this, but I'll tell you. Okay, 1 milligram, 1,000th of a gram of botulinum exotoxin, if it could be distributed to 1 million average size guinea pig animals, would be enough to kill them. 1 milligram of that toxin, if it could be separated and distributed to 1 million average size guinea pigs, would kill all those animals. Okay. There are some important points about exotoxins that you should be aware of. Exotoxins are made of protein. They're often enzymes. Secondly, exotoxins are manufactured by both gram-positive and gram-negative species of pathogen. Since this toxin is a protein, it's going to be encoded by a gene somewhere. And long story short, usually the genes that provide the instructions to the cell to make this powerful toxin are encoded on plasmids, which you hopefully remember. A plasmid is that tiny little extra chromosomal element you learned way back in I guess chapter 4 or so, chapter 3 or 4, encoded by genes found on plasmids and or in genes that are found in bacterial viruses, so bacteriophage. Okay, so encoded by genes found on plasmids and our bacteriophage genomes, which goes a long way to saying it's pretty easy for Mother Nature to swap these genes between cells if Mother Nature is using plasmids or phage particles to do this. These genes get swapped along pretty easily. Finally, because these exotoxins are proteins, most proteins are very highly soluble in blood, which means if you are unlucky enough to be exposed by this protein, this toxin will dissolve in your blood and be rapidly distributed throughout your body every time your heart goes squish and pushes blood out the aorta, right? So are highly soluble in blood, and can be rapidly distributed throughout the body. So that, that's a negative thing, right? We don't want that to be happening for exotoxins that could in fact harm us. Okay, so wow. Okay, I have a question for you. I don't want to spend too much time on the question, but I'd like at least pose it to you so that you're thinking. Here's my question. If a person was sickened by botulinum exotoxin poisoning, could that person's life be saved with massive doses of antibiotic? What do you think, folks, in the chat window? If a person was sickened by botulinum exotoxin poisoning, could that person's life be saved with massive doses of antibiotic? Okay, so take a look at everybody's answer in the chat window, okay? Not everybody's agreeing, and that's what my suspicion was, that you wouldn't agree on this. And here's what I'm getting at. An antibiotic attacks the pathogen. So if the person that's sick is sick with a bacterium that's producing this toxin, the antibiotic will be helpful, but the antibiotic itself does not go after, it does not target the exotoxin that's already in the bloodstream. So the antibiotic can do nothing about the actual toxin in the bloodstream. And if the amount of toxin in the bloodstream is too much, there is no amount of antibiotic in the world that unfortunately is going to save that person's life. That's not going to be helpful. Okay. Because antibiotics target cells, right? The bacteria, and not the toxin, antibiotics won't save this person's life. It's an unfortunate reality. Yes, you could kill the bacteria that might be making additional exotoxin, and that would be useful. That would be helpful. But again, if the amount of toxin already dumped into the bloodstream is very high, the antibiotic's not going to do anything about that, and the person is likely to pass away. Okay. If we had more time, I would tickle you even a little bit more about this. So Samantha, your question, so it can help but not save the person. Well, Samantha, let me put it this way. If the person has acquired the bacterium and has an infection of the bacterium making the toxin, then the antibiotic will be helpful. But if the person was simply somehow exposed to the raw botulinum exotoxin from a bioterrorist, so the exotoxin was put into the patient's body without the bacterium, the antibiotic is going to do absolutely nothing. There'll be nothing helpful there. Okay. Exotoxins. Let's finish off today by just putting a couple. Let's see. Actually, I think I'm going to, there are different categories of exotoxins that your book talks about, folks. I think I'm going to leave that to your capable hands to do a little note taking on your own, simply because I'm running out of time here. Okay. So let me just mention, okay, so in the notes, okay, be sure you read about type 1, type 2, and super antigen exotoxin categories. Okay. And let me just give you a couple notes here in the ending minutes that we have about endotoxins. Okay. Endotoxins. Okay. So, endotoxins are not secreted by the cell. They are actually a physical part of the cell. Okay. So these are molecules that are a part of the cell's own anatomy. And I'm talking the pathogens, the pathogen cell itself, the bacterium in this case. These are molecules that are part of the pathogen cell's own anatomy. They are not secreted like exotoxins. Okay, so let me give you a couple talking points here about this. All right, so endotoxins are made of lipopolysaccharide. Maybe that refreshes your memory. Remember when we talked about the chemical structure of gram-positive and gram-negative cell walls, right? So, folks, make me feel good in the chat window. Here comes a question. Here it comes. Which category of cell, gram-positive or gram-negative, has lipopolysaccharide in its cell wall? Okay, I'm not feeling happy, kid. Okay. 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 Yep, you bet. It's gram-negative, right? So endotoxins are made of lipopolysaccharide, not protein. We took a guess. Everybody took a guess, and that's quite okay. Okay, I appreciate the participation. Endotoxins are part of a gram-negative cell wall. Endotoxins often promote the formation of small blood clots in an animal body, whether that's a human or a traditional animal. And I will mention this. Endotoxins work very similar to the super antigens, which are a subtype of exotoxin. So let me just mention this. They work in a similar fashion to super antigens, which are exotoxins. And by this, what I mean is, if you're unfortunate enough to be exposed to a super antigen, which is an exotoxin, or an endotoxin, basically what these substances are doing is stimulating a chaotic immune system response so that your body basically implodes upon itself because the immune system gets over agitated, and then the immune system caused the majority of harm to the body, leading to potential shock and in some cases even death. Okay, these molecules work by hyperactivating the immune system so that the body's own defenses destroy the body. Okay, you know, that's what's been happening in some cases of COVID patients, where they get this thing that you may remember called a cytokine storm. Yes, Kit. Okay. The problem is that we don't yet have a good way technologically to predict when one person's going to have a cytokine storm and another one isn't, because whether you get a cytokine storm has a lot to do with your personal genetic makeup. And that often is a mystery when you go into the emergency room. We don't know what your genetic makeup is, so we really can't predict it. Okay. All right, folks, it's the end of class. So here's what we're going to do. On Thursday, we're going to tackle chapter 17. I will give you a summary of the major themes and ideas. If I need a little bit more time, we will tumble into a little bit of lab time, but it won't be that much. I promise I'll keep it to a minimum, and we will wrap the semester up. Okay. So any questions right now? Okay, thanks for your energy. I'll see you on Thursday.
Thank you, Professor. Have a good night.
Bye-bye.